Okay, let's go ahead and get started. It is the beginning of the afternoon session. Please grab your seats if you haven't already. So it's really my distinct pleasure to introduce Dr. Hans Clevers, going to give us the plenary lecture this afternoon to start off this session. Unfortunately, Dr. Clevers couldn't be here because of complicated flight arrangements and complicated situation with the FAA. So he is going to be broadcasting to us. Dr. Clevers did all of his training, his MD and his PhD at the University of Utrecht in Netherlands. He then moved to the Dana-Farber for a couple of years to do a postdoc, and then moved back to the University of Utrecht as an assistant professor. And within 2 years, kind of took over the place, became professor and chairman of the Department of Immunology and the Faculty of Medicine, moved up through the ranks beyond that. In 2022, he became, well, before that, he was chair of the Department of Immunology, director of the HUBRECHT Institute, which is part of the Royal Netherlands Academy of Arts and Sciences. He then I think has taken a step out, but I think maybe with only one foot into pharma. He's head of pharma research and early development at Hoffmann La Roche in Basel, Switzerland. And I think he's still the interim director of the Roche's Institute of Human Biology. And you can see in your program, he holds more appointments than I've actually mentioned so far. So as many of you know, you've heard his name mentioned already in a couple of talks today. Dr. Clevers is recognized for his pioneering studies of the Wnt beta-catenin signaling pathways in disease models and stem cell biology. I think his early focus was in the role of this pathway in cancer cell biology, and he provided a very seminal description of the molecular explanation of the initiation of colon cancer. Subsequently, he established the role of the Wnt signaling system in adult stem cell biology. He was the first to link the Wnt signaling system with adult stem cell biology when his group showed that disruption of a transcription factor, TCF4, that he had previously discovered leads to the abolition of the stem cell population in the crypts in the small intestine. So amongst the Wnt target genes in the intestine, he found that the LGF5 gene that you've already heard about was unique in that it marked the stem cells at the crypt bottoms. And he subsequently established that LGR5 expressing cells represent the stem cells in the small intestine and the colon and the hair follicle and the stomach and in many other organ systems. I think he was also responsible for establishing that LGR5 is the respondin receptor, which we also heard about earlier this morning. And his group pioneered LGR5 respondin-based culture systems that allow the outgrowth of single mouse or human stem cells into organoids. And organoid biology is really where I know his work from, because this is not my area. My area has been in epithelial biology, and the revolution that this type of technology has made in the field of cystic fibrosis, where patient cells in a relatively non-invasive assay can be put into culture conditions that Dr. Clevers has pioneered to not only grow out and understand the physiology of those tissues from the individual patient, but to actually explore the responsiveness of that particular patient's cells to putative molecular therapeutics. I've seen this in action in the CF field, and this is going to be true in many other organoid systems that he's developed. I don't want to spend too much more time. I've actually learned a tremendous amount from reading Dr. Clevers' biography, CV. He's published over 800 peer review articles. He's been cited nearly over 200,000 times. I think he is going to focus his talk today on the organoid technology that we've heard a little bit about today. So without further ado, Dr. Cleavers, I'm glad you can still join us, and we look forward to your presentation.
Yeah, so I hope you can hear me. I'll start sharing my screen. There we go. Yeah, first of all, let me apologize for not being there at Bob's symposium in person, but I was in Toronto. I saw all the drama at the US airports. I realized I had to take 3 short flights on 3 consecutive days, and I thought this is going to be a disaster. So I decided to fly back home, and I give this talk virtually. So what I'll do, I'll probably provide some slides to what we just heard about what my lab has done in the past 30 years or so. Actually, we started as immunologists, and I was going to clone a transcription factor in my small lab. We did one. We cloned TCF1 for T cell factor 1. A number of years later, we realized that these TCFs are the endpoints of the Wnt pathway. So that took us away from T cells to developmental biology. We had flies and frogs and things. And eventually this took us to this organ, the gut. You're looking at the villi. Probably you might have seen them already today on other slides. And these structures are called the crypts of Lieberkühn that were described 3 centuries ago by Johannes Lieberkühn. And about a century and a half ago, people realized that crypts and villi are connected. And then a little bit later was found that these, near the base of the crypt, there should be stem cells that are remarkable. And their identity remained unknown for quite a while. So this is a real view with a confocal on small intestines, probably mouse. So you see villi. In green are the most abundant cells, the enterocytes. These take up the nutrients. So basically, they perform the single important function of the gut. In the red, you see goblet cells. They produce mucus such that the contents of the gut can move smoothly over the surface. Sorry. And this is a different view of the same organ again. So you see some villi in the back. This is a villus here. It's probably about 6 or so crypts that surround the base of villi. And what you see here is 2 of these crypts. Near the base, these large cells, I'll show them in better views, are called Paneth cells, discovered a century and a half ago. And somewhere near the base of the crypt, there should be stem cells. And people believed 15 years ago, 20 years ago, that these cells here, so the bottom of these brownish cells, would be the stem cells. What was clear that whoever produces the daughter cells, these daughter cells proliferate very rapidly. Cell cycle times of about 9 hours or so. While they move up, it takes them about 2 days from here to here. Then they differentiate out in a number of different cell types that I'll show. They continue to move up the flanks of the villi, and now they are exposed to the gut lumens. Probably an important factor why they are so short-lived, because about 5 days after they're formed at the base of the crypts, they reach the tips of the villi and they die. So this was seen by radiolabeling of DNA probably in the late 40s of the last century. And it's really a conveyor belt that we're looking at. There's one cell type that escapes, or 2 cell types that escape this flow. Paneth cells, basically, when they form, they turn around and they settle at the base of the crypts. They live longer. Stem cells presumably live lifelong, but all other cells live 5, 6, 7 days or so in a mouse gut. So our entry in this field was essentially the fact that we had cloned these TCFs, the endpoints of the Wnt signaling pathway. We knocked out TCF4, as mentioned. When we did that, the mice died, and it took us a long time as immunologists to find the reason why they died. And actually, this process comes to a halt. What happens then is the villi very rapidly shrink to almost nothing, and then the mouse dies. So they basically die 5 days after Wnt signaling is blocked in the crypt. So this was indeed the first link, I would think, of the Wnt pathway, not only controlling development, but also playing a role in adults. And that is for stem cells. Probably now I would believe that most, at least epithelial stem cells, thrive on Wnt, as I'll show you a bit later. So Wind drives normal stem cells, and about the same time, almost 3 decades ago, together with Bert Vogelstein, we showed that using our TCF, our wind reporter assays, that in colon cancer, where APC is typically lost, beta-catenin is up very high, the effector of the Wnt pathway, and beta-catenin TCF transcription is very, very high. So on the one hand, you have a physiological dependence on Wnt of these stem cells, and then in the same organ to create cancer, you almost always will see activating mutations in this Wnt pathway. So we had these two observations. This is now 97, 98. There were no good technologies yet to solve transcriptional programs at the genome level. Even the genome wasn't known. But then Pat Brown invented DNA arraying, and this allowed us, I won't show you the experiments here, but this allowed us to determine what genes are controlled in crypts by Wnt signaling. And we found actually they are the same genes that are controlled by the same pathway, but then mutationally activated in cancer of the gut. A list of about 50 or 60 genes. So we worked our way slowly through this list. We believed that the stem cells, that was what the common knowledge was, would be somewhere here. But then we stumbled across, and we is in this case Nick Barker, British postdoc, who is now a professor in Singapore, stumbled upon this gene LGR5, a 7 transmembrane receptor, that lit up these tiny cells that we had entirely missed for a number of years. So these large cells, again, are the Panner cells, but these little stripes are LGR5 positive cells, slightly bigger than a nucleus. And this is actually a mouse in which Nick knocked in green fluorescent protein. So that's why we see them in green. They also they express a Cre-ER. So this now allows us to, at will, for the non-molecular biologists in the audience, at will to turn these stochastically blue, and we give a very low dose of the inducer, so we only hit an occasional potential stem cell. Nick leans back, and then over the next 2 days, he saw proliferative daughters moving up. After 2 days, the first blue cells were exiting the crypts as predicted. They matured into the different cell types. Actually, we saw all of the cell types of the gut in these blue ribbons that are formed by this one cell that sits here. This process continues, so now they move up the flanks of the villi, now working really hard to keep everything bad that's out here in the gut, keep it out of the body, but pump nutrients and liquids into the blood and lymph vessels. And when they're about 5 days old, the first blue daughters arrive at the tips of the villi, as predicted, and they die, they undergo anoikis. So if we don't analyze the mice after 5 days, but we leave them for 2 years, we will still see these blue ribbons, implying that the cell that Nick marked 2 years earlier is essentially a long-lived cell, unlike all other cells in this tissue. And second, as I already said, in these ribbons, here we see all of the different cell types, goblet cells, enterocytes. I'll name a few more a little bit later in the talk. So the cells are long-lived, they are multipotent, they make all of the other cell types of the tissue. So we propose these cells as being the stem cells of the gut epithelium and being these extremely active stem cells. There was a lot of resistance in the field because there was a strong belief that stem cells rarely divide. It's essentially their only job is to create daughter cells, but still it was felt that cell division could lead to mutations, deleterious mutations, so you'd lose your stem cells, or maybe oncogenic mutation, you would create cancers. So essentially, when people are looking for stem cells in tissues, they were looking for cells that were rarely dividing. So you label the DNA, you come back a month later, and the cell that hasn't divided in that month will still have its DNA label, and that was then a candidate stem cell. So these definitely do not fit that bill. They actually divide every day, in the mouse every 23 hours. Now, a more elegant way of doing the same experiment was set up by Hugo Snippert and Laurens van der Vlier. They obtained the rainbow cassette from Jeff Lickman at Harvard. They built it into a knock-in mouse. This now allowed them at will to basically let each of these stem cells, probably about 10 or so, select an individual color from this reporter that you see in the slide. And then our animator, Jeroen Huybe, believed that this would be the outcome of the experiment. We said it was a bit over the top, but then we got the real results. So muscle would be here, the food would pass by from here to here, and you get these nice villi. And if you zoom in, indeed, individual crypts always resolve into a single color. So there were 10 different stem cells. Actually, we can mark them in 10 different false colors. But if you wait 2-3 months, you come back, typically a crypt has become clonal. This one has become red, this one has become blue, this one is yellow, green, and you see that they keep on pumping out daughter cells in the color of the crypt. So this is essentially a village, and you get these beautiful zebra stripes that run up the flanks of the villi. Well, two important conclusions from this experiment. First of all, the stem cells that sit here essentially don't divide asymmetrically. That's what your textbooks tell you. A stem cell divides, it makes a new stem cell, and it makes a daughter cell. What we see, and we also have movies of this, of live movies in mice, that the stem cells, if there's about 10, they divide. By the end of the day, there would be 20. However, there's only space for 10, so randomly they kick each other out. And if you start with 10 colors, you would have two reds, two blues at the end of the day, but there's no space for all of these cells. They get kicked out, and sometimes by chance the two red ones are gone, and then that color is lost from the crypt. And then eventually this game is played every day. After about 2 months, you typically see that one of the stem cells has won. It was not a better stem cell, it was just luckier than the other ones. And we see no asymmetric divisions. They are symmetric divisions followed by a neutral competition for space. The other observation was that this was not only happening in LGR5 cells in the gut. We started realizing that LGR5 probably marks all epithelial stem cells in their active state, so hair follicle stem cells, stomach stem cells. Liver is negative, but when you damage the liver, the stem cells wake up and you'll get positive cells in the liver. And it again confirmed that these cells are constantly dividing, at least in the mouse. As already mentioned, we found out much later what LGR5 does, but just to give it away now, it explains the experiment a bit better that I'll show you. So this is the classical Wnt receptors, frizzled. There's 10 frizzleds, 7 times membrane receptor, LRP 5 or 6, single pass membrane receptor. So these are the co-receptors for Wnt. This is frizzled. So if you're a stem cell, you'll have frizzled and LRP, but you'll also have LGR5. And here you see, Wnts are lipid modified. They don't really travel far. They're typically produced by a direct neighbor. When Wnts activate by bringing back together these two receptor molecules through beta-catenin and TCF, they activate target genes. And amongst those target genes are two E3 ligases. You see the names here. They're actually tumor suppressors. In a very dedicated way, they ubiquitinate frizzled and remove the active receptor from the surface. This is great for development because this happens within hours. So 2 hours after the signal starts, it's gone. This negative feedback loop, not good enough for stem cells. Stem cells need a really robust, prolonged signal. And this is where LGR5 comes in and its ligand are spondin. So when the E3 ligases appear in an actively Wnt signaling molecule, LGR5 binds our spondin with one end, the other end of the molecule binds the E3 ligase, removes the E3 ligase, so it essentially breaks this negative feedback loop. And now the Wnt complex that's signaling here, frizzled LRP, continues to signal. So in the presence of LGR5 on a cell and our spondin from the medium, from the niche, we read out strength of the Wnt pathway that are like 100 to 200-fold higher than what you would read out with Wnt alone. So Wnt alone is great for development, but Wnt plus our spondin to block this negative feedback loop is really what the stem cells need. So we didn't know this when we first of all found LG5 as a marker, but it explains why it is expressed on stem cells. We also didn't know how our spondin works, and I'll get back to that. But that was also found later after we did a crucial experiment that we didn't really understand too well. So I'll give you an overview of what the LGR5 stem cell does. So there was an old name, LeBlanc in Canada, had actually seen these cells by EM. He called them crypt-based cells or crypt-based columnar cells. We now know that they make a significant number of different cell types. The absorptive lineage, enterocytes I showed you, so they essentially perform the gut function. They take up nutrients. About 90% of all cells, 95% are enterocytes in the small intestine, colonocytes in the colon. In the small intestine, they're responsible for taking up amino acids, lipids, sugars, etc., etc. In the colon, their equivalents only take up water. There is a strong amplification to this end. So the transit amplifying cell here divides 3 to 4 to 5 times while it decides to become an enterocyte. or an M cell. If I have time, I might say something about that. These are dedicated cells that don't sit on the villi, they sit over the so-called Peyer's patches. These are lymph node accumulations in the gut epithelium. Actually, typically, there are lymphocytes inside this hole here, or dendritic cells. They are presumed to take up particulate matter from the gut and then transport it to the underlying lymph tissues in the Peyer's patches. BEST4 is a cell that doesn't exist in mouse, is recently discovered by single cell sequencing. It's about 1% of all the cells in the epithelium. They are responsible for maintaining electrolyte balance and liquid balance and pH in the gut. I won't talk about them. And on the left, there is a tuft cell, probably well known to many of you. For instance, important to drive out worms from the intestinal tract. Goblet cells make mucus. They also make bactericidal proteins, so they also help protect the gut. Paneth cells in their granules have lysozyme. They have defensins, also protect the gut. So essentially, there is 1, 2, 3, 4, 5 cells that are involved in immune protection. M cells actually, because they transport antigens. BEST4 cells, they create watery diarrhea and drive out bacteria when you have a bacterial infection. And then there's this group of cells, there's 6 of them, that has, and I'll say more about these, have become very, very interesting in recent years because they are the cells that make hormones. They sense things in the lumen. They have taste receptors. They have also all kinds of other receptors, not exactly know what they respond to, but then when they sense their ligands, they will then secrete hormones into the bloodstream. So this goes into the gut, but this goes into the bloodstream. 6 cell types. The most famous one is the L cell. L cell makes GLP-1, and GLP-1 is essentially the inspiration for all of the weight loss drugs, Mounjaro and Wegovy and Ozempic, that are almost daily in the news. But there's many other hormones and also hormones that are poorly characterized that are produced by these hormone secreting cells that line the gut and apparently convey information from what's in the lumen of the gut to the body and control satiety, insulin secretion, etc., etc. All made by the very same single LGR5 stem cell. Now, I told you that these stem cells constantly divide, and actually it was then, first of all, that was against the view on how the stem cells should behave. Also, there was this strong belief that wild type cells, normal cells, if cultured, they will rapidly die. Primary hepatocytes is a good example, I guess. So you have to do your experiments fast on an explant or something like that. The cells that continue to grow out are cancer cells and cell lines. All cell lines are cancer cells. When you transplant them, they make tumors. So, but seeing that our stem cells were dividing possibly 1,000 consecutive times in the mouse gut, the same stem cell, I sat down with Toshi Sato, a gastroenterologist from Tokyo, who's now a professor in Tokyo and with his own lab there, very successful. We sat down and we thought, well, maybe we can rebuild the crypt environment. And we knew quite a bit. I already told you that our spondin is a Wnt amplifier. I showed you the mechanism. We didn't know then how our spondin worked, and that LGR5 was the ligand, the receptor of our spondin. But we decided to use our spondin rather than Wnt in our cultures. Wnt is lipid modified. Actually, then no commercial source of Wnt worked in our hands. You could buy it, it was very expensive, it didn't work. We could make some Wnt by ourselves, but it is lipid modified, it sticks to everything. Nuvello, who was trying to develop our spondin as a regenerative medicine drug, went broke and then gave us a very large batch of our spondin, which is why we actually used it. It's easily produced, it's soluble. And we now know it was the ligand of LGR5, and it is a Wnt amplifier. EGF, known as a mitogen for the gut epithelium for several decades. Noggin, we knew from a transgenic mouse experiment, if we express noggin in the intestinal lining of a mouse, what we see is we get many, many more crypts, and the crypts run up all the way the flanks of the villi. So somehow blocking BMP by a protein noggin that binds BMP factors, you create a crypt permissive environment. Now, we hope to see a lot of stem cells, but what we got were these structures. And then when Toshi looked carefully, he realized that these structures have a central lumen, and these central lumens are lined by all of the differentiated cell types. I'll get back to that later. And these buds turn out to be crypt equivalents. And I think, yeah, and so what you see is actually they're round, they don't really look kind of good, but when you put them in collagen gels in bundles, they will fuse, and we can make these long tubes. This is our effort at bioengineering, and we are staying for lysozyme here. These are Paneth cells. So indeed, this is a tube like the gut, and these things that stick out are Paneth cells in crypts of Liverpool. So really remarkable that a single stem cell without any architectural cue, just 3 recombinant proteins, no serum. I should stress we do it in Matrigel. This was Minna Bissell who told us that we should do that. Matrigel is a mix of laminin, collagen, and other extracellular matrix proteins. I'll get back to that towards the end of the talk. That was the first surprise. So they basically make a structure that really looks like a gut. It has all of the cell types of the gut. Also, they never stopped growing. We've now had them in culture for 10 years or so, whereas mice only lived 3 years. So despite the fact that we couldn't see any emergence of oncogenic mutations in these things that were growing, we asked, well, are they really normal? So Toshi hooked up with his former colleagues Mamoru Watanabe and Tetsuya and Shiro in his lab. Toshi sorted a single stem cell from our GFP, LG5 GFP mice, 1 cell, put them in the culture medium that I just showed you. The mice were also marked with RFP. So green means you're a stem cell. There's one here. Red means you come from this mouse, because every cell in the mouse is red fluorescent. So he went from 1 cell to about 100 million cells, froze them, sent them to Tokyo, where Mamoru's people had treated mice with DSS, which induces an inflammatory bowel disease-like syndrome, very popular in gastroenterology research. They infused these red fluorescent organoids with the green stem cells into the colons of these mice. Their basal side is on the outside, so integrins are on the outside. With these integrins, they can engage with exposed collagens when there is an ulcus, as you can see here. They then merge together, and like a living band-aid, they cure these mice. So this is, of course, not the real experiment that is here. You see the colon of one of these mice. This was apparently a region where there was an ulcus. In orange, you see the red fluorescent Dutch tissue in this black non-fluorescent background of the mouse. The only way to find these patches by microscopy is looking for RFP expression. So here you see one of these patches, but by everything, H&E or marker staining, this is fully normal tissue. So we never saw any neoplastic, so no polyps or anything. Also, another prediction would have been these cells, because of the Hayflick limit, must be exhausted. They've been massively expanded from 1 cell to 100 million. You'll probably lose these patches. That also didn't happen. So telomeres stay long in these organoids. Organoids grow forever, but also when you transplant them, they make healthy tissue. And based on this, Mamoru has gone on to transplant human to mouse and is, as I understand now, in the midst of a trial where he transplants human to human. So I already told you that LG5 is not only a marker for the gut stem cells, but it looks like every epithelium has wind-driven stem cells, and when they're active, they express LGR5, and they use LGR5 as a co-receptor for wind. You can see a long list here of tissues that we or other labs have been able to turn into organoids. And I guess this was a paper of about 6-7 years ago for taste buds that probably interest many of you. We've used this now for many different things, personalized medicine, but also basic, gene function studies. CRISPR works very well, as I'll show, but you can model cancers directly from patients, or you can create cancers with CRISPR. Cystic fibrosis, you can create directly from patients. And we've done a number of infectious disease studies. Flu, actually, you can easily show if a flu is really pathogenic or not, a particular flu strain. With COVID, we could show that chloroquine does not work. It works in the cell lines, but it does not work in organoids. And there's many other applications, I guess. So from our mouse experiments mostly, and then also from our organoid experiments, we could actually, we've sort of solved the rules by which a stem cell, so now the stem cell is at the top of the hierarchy, by which it actually can make all of the different cell types. So you see basically one absorptive cell type enterocytes. There should be M cells here and best 4 cells. We also know what to do, but wind on, notch on, you have a stem cell. Wind off, notch on, you make an enterocyte. notch off, you make a secretory progenitor, and this directly can differentiate into goblet cells, into one of the many different hormone secreting cells. You see here the L cell that makes CLIP1, and then of course the tuft cells are also somewhere in this pathway. I'll say more about the enteroendocrine cells now. So the classical nomenclature essentially names the cells after the hormones they secrete. So N cell are neurotensin expressing, ECs express serotonin, D cells express somatostatin. They express more hormones and there's some overlap, so it's not really a black and white phenotyping. And as you can see, these hormones control motility, appetite, insulin secretion, mood possibly, etc., etc. So here you see again what we're studying, crypt-based. So this is the LG5 cell that can make other secretory cells, absorptive cells, and the 6 EECs. In the past few years, we've given some attention to the biology of making EECs. So we don't really know what drives a secretory progenitor to become an enteroendocrine cell. We have not found very specific signals for most of the other cell types. Sometimes it's an interleukin or a ranked ligand that uses M cells. For EECs, they appear in our organoids or in mice. We don't really why. Also, we don't know what sets which subtype they make. There's really regional specificity. The ghrelin cell, for instance, sits close to the stomach. The L cell sits close to the colon. Because there's no cell lines and mice are quite different from humans, it was interesting to study EECs both in mice and in humans. And here you see, this is a mouse organoid where we mark chromogranin A, which is a pan-enteroendocrine marker in green by a knock-in. You can nicely see there are enteroendocrine cells in these organoids spontaneously arising there. And then Helmut Gehart came up with a very smart, that I hope I can explain, a very smart strategy to capture early and developing enteroendocrine cells in mice. And what he did is he realized that neurogenin 3, which is essentially the transcription factor that pulses, you see that here. So when you're a secretory progenitor, you're here. When you then undergo a pulse of neurogenin 3 expression, it's just a few hours, you're then destined to become one of the many EEC subtypes. And what Helmut did is he knocked into the neurogenin 3 locus a rapidly folding, here we go, a rapidly folding green protein that is highly unstable. He made it unstable, so this is neon green. So the idea would be the moment neurogenin 3, this peak happens, you'll see almost immediately a green peak that also then rapidly disappears. Tomato is a slowly folding, slowly maturing fluorescent protein with a much longer half-life. So from the same locus, where you first make a green protein, you then make a red protein. And this is basically allows us to follow, just by following the shift from green to red, to determine the age of a cell after the moment that it decided to become an enteroendocrine cell. Of note, I already said this, but we don't know what induces this pulse. We'd really like to find out what tells this cell to activate neurogenin 3. Is it just a stochastic event, or is there some external signal that tells these cells to become EECs? So on the left here, you see an organoid. This bright green is autofluorescence in the lumen, but the epithelium lines the lumen. And if you follow, for instance, let's wait till it loops. If you follow this cell here, it's green, red, becomes green and red again. So it essentially goes through of these cycles, and many of these cells do this. And when we know how to increase generally more enteroendocrine cells, essentially by taking EGF out of the medium, and then indeed we see many more of these cells. But you might again see, I hope that you can see this, the pulse, green, red, green, red. So they actually go through of these induction cycles. If we don't know, we think maybe this determines which subtype they'll become, but we don't know that yet. But anyway, so this thing works in organoids. And what Helmut then did, and this again summarizes, hopefully, so we delve into the crypts of Lieberkühn. There are pennant cells and stem cells. They're in a very regular pattern. So this is a common secretory progenitor. It decides to become an enteroendocrine progenitor. What it now does, it upregulates neurogenin 3, as I told you. At the same time, it starts producing those two proteins, but one of them is rapidly fluorescent but then disappears rapidly. The other, it takes a while before it becomes fluorescent. You see that happening here, but then it stays red much longer. And just by reading out intensities and relative intensities, and actually that's what we noted, that in crypts, you'll see green ones deep down, yellow ones halfway, and red ones on the top. Now, we can now directly sort from the mice individual EECs. We record their color, so we know their age after the neurogenesis repulse. We then do a single cell sequence, and now we get not in sort of calculated, modeled age, but this is really actual measured age. We can now fill the EEC hierarchy with single cell signatures starting from green to yellow to red cells. And then hopefully we can now basically draw the entire tree, and we know for each of the cell types now their entire gene expression patterns, including the receptors that they use to secrete their hormones and the hormones that they produce. So Helmut then came up with the following. So here are the 7, 1, 2, 3, 4, 5, 6. I have to show you a little bit more. There's another slide. So there's 6 enteroendocrine cells. He focused for this paper. He focused on transcription factors and actually knocked out quite a few. We can do it quite easily in organoids, and then found out that about half of these transcription factors would have a very significant, if not an absolute effect on the formation of a particular lineage. I must say that after having done all that work, it's a little disappointing because we already knew that transcription factors do this, they set fates, but we really like to know what actually controls the progenitor to move into the delta cell or the X cell or the L cell fate, and maybe we can manipulate it if these are exogenous signals. We can do it for all other cell types now in the gut, but not yet for the EECs. Also, we found that ECs are a little bit different. They make serotonin, so a small molecule rather than peptide hormones. They only take a day from their precursor, but these cells all take at least 48 hours to form, to become a mature cell from their precursor. Then, just summarizing a study by Hugh Böhmer, we learned that cells at the base of the crypt, they live in a blocked BMP environment. So there are BMP inhibitors secreted. That's also why they are in our organoid cocktail. But when we, in the organoid cocktail, replace the BMP inhibitors by BMPs, we actually create a gradient that exists in the crypt villus. So this is a BMP gradient, high levels BMP in the tip of the villus. What we notice is actually that K cells, or let me take the example of L cells, at crypts have a very different gene expression profile from the same L cells when they move up this gradient. So they might hang around here for a few days, they then move up. When they hang around here, they make, where is it? GLP-1 to high levels. But the same cells, when they move up, they switch GLP-1 down, and now they express neurotensin. And you might recall from one of the first slides, probably not, but it was in the slide, that actually there was an N cell, which was a neurotensin cell, and there was an L cell, which was the GLP-1 cell. But we now know it's the same cell that basically lives in a BMP blocked or a BMP active environment. Enterocytes do the same thing. They take up amino acids here, and when they move up under the control of BMP, they switch to lipid transporters, and here they take up lipids. And goblet cells also do the same thing. They move through this BMP gradient, and they mature while they move up. I don't show you any data, but just wanted to show you where we now think there are 6 enteroendocrine cells. In the papers that Joep has published since, also will publish independently in his own lab in Basel, you're probably interested in the receptors and the transporters that they have. You can find them in the papers on these enteroendocrine cells. Because of the setting, I also try to say a little bit about taste buds. So, of course, protocols were published. We have, here it is. We have also attempted to grow organoids from taste buds, and particularly from, in this case, from the posterior taste bud. And what Laurens Verwey and Suk-Yong Kim in the lab have recently published is that actually we realized that there's always a little salivary gland, a von Ebner gland, near a taste bud. And we thought, well, maybe they are somehow related through stem cells. And actually, there's an LGR5, LGR6 stem cell that drives taste buds. That's published by Renadal, not by us. But we also realized that salivary glands use the same stem cell. So we asked ourselves, is it possible that there is a hub of a stem cell that can either decide to go up into the taste bud or down into the salivary gland? This is really a functional unit because the flow of saliva probably allows, I'm not an expert here, but probably allows tastants to then come in touch with the receptors that sit on the taste bud cells. SOX2 is a marker for early taste buds. You see there's SOX2 in these structures. LGR5 GFP sits here, and SOX9 sits in the salivary gland. And if you put this together, it looks really like a continuum, and other people have described it. So we ask ourselves, is there a single stem cell that fills both compartments, the salivary gland and the taste bud, or are they separate cells? And of course, we can do this in organoids. And what the guys made is basically from Nick's LGR5 GFP mice, sorted individual LGR5 cells from the back of the tongue of these mice, single clones, single cells, as Toshi originally done. So we know we actually start from one stem cell, we then grow them up, and we can induce them to differentiate. And here you see 3 of these clonal lines. And in essence, in organoids from these posterior taste buds, in the clonal lines, we see the LGR456 precursor. We see where are they? The taste and the saliva, they actually group together here, and we see myoepithelial cells. So I think, where is this? No, so I should probably go back here. I think this shows that you can actually take a single cell from the stem cell region here. And then depending a little bit on the culture conditions, you can make organoids, but then you can, in those organoids, find, again, you can push them in one or the other direction. You can both make taste bud tissue and you can make salivary gland tissue. They also went on to look at the anterior tongue. This is, of course, the first beautiful paper on salivary gland taste buds, PNAS. And this is the anterior tongue. I don't think that there were protocols for anterior tongue yet, but they managed to also do this. And in the anterior tongue, they find in the organoids, I don't give you much data here, I don't have enough time, but we find Merkel cells and we found taste bud cells. And then there is, again, a common stem cell that makes taste bud, and in this case, the Merkel cells. Also, this is ongoing, but we are trying to do the same thing in humans. We've not seen, maybe we missed them, but data sets of human taste bud single cell data. And so we focused on the circumvallate papillae and tried to turn them into organoids. This is a single cell sequence from human tongue. You can see there's many cells, but they're mostly immune cells and keratinocytes. So this is all essentially squamous epithelium. But we do have taste bud cells, a few hundred here. And essentially we can map them back quite well on the mouse single cell data, although there are clearly significant differences. So we now know what these human taste bud cells look like. And when there's LGR5 cells here as well, we've managed to grow organoids. And in those organoids, we start seeing also the various types of human taste bud cells. This is ongoing, but it's to show that we actually do make an effort to do this in man. Generally, it's usually easier to grow mouse tissue. It grows faster. Cell cycle times are shorter, but also typically we need to add a few more small molecules or growth factors than we do in the mouse to be able to grow them long term and make real, so be able to, for instance, to make CRISPR subclones. Finally, organoids are grown in 3D. The way it's done, so matrigel is liquid at 0 degrees and is a gel at 37 degrees. So this is you buy it as a liquid on ice, you put cells in, you then drop these, so these are called domes, on the base of a plastic dish, put them in a 37-degree incubator, and then you have your hydrogel with the cells floating in here. You then basically add the growth factor cocktail medium, no serum, typically 3, 4, 5 growth factors, depending a bit on which tissue you're growing, always a Wnt activator, always a tyrosine kinase receptor activator, and always a BMP TGF-beta inhibitor, and then maybe some hormones like estrogens depending on your tissue. Matrigel is magical. I don't think there's anything that at least we have tried for ourselves or from commercial sources or from collaborators that matches what Matrigel does in organoid culture. However, it's very expensive. It's made by growing a mouse cancer sarcoma line in the abdomens of mice, not allowed in the EU anymore. Extracts from what's growing in the bellies of these mice is then purified, and this results in something that's strongly enriched for laminins, collagens, fibronectins, but it's not pure. There's large batch-to-batch differences. There's all sorts of unknown components in this extract, and this will probably never be compatible with clinical use of Matrigel. So there's a strong push everywhere to see if you can come up with something that's more standardizable, cheaper, and more reliable than Matrigel, and not animal-based. So what Matrigel essentially provides is it basically looks to the cell, to an epithelial cell, like the basement membrane that epithelial cells typically sit on. So epithelial cells have integrins here. They bind to collagens, laminins here. This creates a signal. The signal allows the cells to polarize. They know, okay, this is my basal side and this is my apical side. But more importantly, it stops the cell from killing itself and undergoing anoikis. So an epithelial cell that doesn't see basement membrane will almost immediately die. It's the integrin signals that keep it alive. And that's probably, or very likely, the magic that Matrigel can produce, but that actually no other hydrogel currently has produced. So a little bit about integrin signaling. A lot of this was solved a long time ago in the 90s. So integrins sit on many cell types, immune cells, epithelia. They typically sit in an inactive state. You see that here. It needs an inside-out signal, some activation signal, to open the complex up. There is an alpha chain in blue. There is a beta chain in green here. The beta chain is believed to be mostly responsible for signaling. The alpha confers most of the ligand specificity. There's many alphas, there's a few betas, and the ones I'll be talking about is beta 1, very important on organoids. And then alpha 5 and 6 are very important on organoids. Now, once it has reached this state, it can now bind, for instance, to laminin. And if it does so, it creates a bit of a signal from the beta chain. If then the integrin complexes get clustered, of course, like collagens have multiple repeat motifs, laminins form large networks, so this is not too hard for extracellular matrix to accomplish. You then get a much stronger signal. This now strongly polarizes the cell, lets it to form junctions, for instance, but particularly doesn't allow the cell to kill itself. So we've looked at lots of things. The RGD peptide is widely used. It's a small fragment from fibronectin that engages alpha 5 beta 1 a little bit, not good enough. If you put it on gels, if you functionate gels, it doesn't really replace Matrigel. You can maybe grow an organoid 1 to 2 passages, but then it fizzles out. So we looked everywhere, and then finally Joost Wijnakker realized that there is a bacterium, Yersinia. You probably know Yersinia pestis, the cause of the plague. There's other Yersinias that cause chronic inflammation, chronic infection of the gut. And what these Yersinias do, they exploit a very unusual characteristic of M cells. So these are villi in the background. This is a Paris patch. So epithelium comes from the same stem cell, but the ranked ligand has told the cells to become M cells. M cells have a hole, and typically there's lymphocytes in here. But for this story, it's very important that they express their integrins. They have integrins, but they're not on the basal side. They are on the luminal side. Nobody knows why that is. It's probably the only cell that does that. But that's what Yersinia exploits to get into the human body. So Yersinia sits in the lumen of the gut, ingested with food. Here it is. What it now does, it has these molecules on the surface called invasin. Invasin, it has a 20K fragment here on the extreme C terminus that can bind alphas and betas, bring them together, activate them, and now the M cell is forced to transport the bacterium through the cell into the underlying tissue. And now basically Yersinia has succeeded in penetrating the body of a mouse or a human being. And we actually now know that this is done by invasin. Now, Ralph Isberg in Tufts in Boston has worked on this, published beautiful papers in the 90s in all the top journals. Actually, this is sort of forgotten. There were no citations of this protein in the past 3 or 4 years. He also solved a structure. So this is the extreme C-terminus that sticks out of the bacterium, extremely stable. It sits in the lumen of the gut, so it survives everything that's in the lumen of the gut. Very easy to produce in E. coli. And this is the part that's by itself enough to activate integrins. Now, invasin binds a number of different alpha beta, so they're always beta 1, but there's alpha 5 beta 1, the fibronectin receptor. Everything in the red box binds to invasin. Collagen receptors do not bind invasin, so alpha 1 and alpha 2 beta 1, but importantly, alpha 6 beta 1 is the laminin receptor, and that's the key we feel for Matrigel and also the key for growing organoids. So Joost wrote to Ralph Isberg, and Ralph had a very old batch, 25 years old, but he said it's a very stable protein, it might still work. So we got a tube from him, and Joost did a very simple experiment. He, in 2D rather than in 3D, he just coated protein on plastic, 10 wells, no coat, there was no growth. Bovine serum albumin, there was no growth. Matrigel, BME, there's okay growth. Invasin, the very first try, 3-4 times faster growth on this coat than on the Matrigel. And here you see one of these organoids that he tried. This is a human intestinal organoid that carries the Fuji reporter, where G1 cells are red, G2M cells are green, and S-phase cells are yellow. And you can see in a matter of a week how rapidly this grows as a sheet. So it's not an organoid, we call it organoid sheets. If you look carefully, this is a two-dimensional sheet grown on Matrigel BME. And this is the same sheet grown on invasin. You might see that actually polarization is much tighter. Actually, when we measure transepithelial resistance, is much higher on invasin than it is on BME. And here you see a much larger sheet. See, the cells are beautifully polarized. So invasin sits in the base here on the plastic. This is the luminal side. And you see a lot of enterocytes with their brush borders, for instance, in green here. And then we made a reporter, I haven't shown it yet, a while back. Goeiewege and Lin made this reporter 2 years ago. So essentially with CRISPR, they knocked in a green fluorescent protein in the mucin 2 gene. Goblet cells are green. Pannet cells are with a reporter for the ventin A5 are orange, and the hormone secreting enteroendocrine cells that I already showed are purple. This is a top view comparing Matrigel in 2D and invasin on 2D. If anything, this looks better to us. But also if you look, for instance, at the specific cell shapes of these enteroendocrine cells, they're like swan shaped. That's what you see also in primary tissue. So there's a little tip that sticks in the lumen of the, this is where the food would be, where they pick up their signals. And then the cell body sits here. This is where the hormones sit, and they secrete. If they would secrete, they secrete their hormones into the bloodstream here. Goblet cells do the opposite. They have an opening to the top because their mucus shouldn't go into the bloodstream. It should go into the lumens of the organoids, the lumens of the gut. And there's a few Pannet cells that do the same thing. We've actually grown organoids for about 20 passages in 2D on invasin. When we then compare them to the starting single cell readout, we see similar ratios of the different cell types. We see similar maturity. Of course, most epithelia in essence are 2D structures. They grow as a sheet, and then the sheet is folded in space. So in the gut it would be villi and crypts. Many epithelial assays, transport, etc., are 2D assays. So also think about drug development. I've been at Roche, so none of this, I should state, is done at Roche. It's all done in my academic lab in Utrecht. But I've seen how industry struggles with these very complex, hard to standardize, technically complicated assays that involve 3D organoids. In 2D, you can automate. We can do it in 384 well plates. And many of the assays are essentially well known and well established for 2D. And for 3D, they don't exist. So we think this is really important. Tim Springer, our collaborator, who has worked on integrins for 4 decades or so, he created a structure. Saket and Ying did this. So invasin bound to an alpha and a beta chain, in this case, the fibronectin receptor. So it engages the very same amino acids that fibronectin would do, the RGD peptides, but the contact surface is larger. Probably that's why it's more effective, and the affinity is a bit higher than for the natural ligands. But this basically underpins why invasin does what it does. Tim also generated an alpha 6 beta 1 structure where the two domains of invasin are slightly turned relative to each other. So it looks like invasin has really been evolutionarily designed to interact effectively with a number of different invasins. We can even grow reptile organoids on invasin. So this is a snake venom gland organoids, implying that this protein takes advantage of a very evolutionary conserved surface, namely the alpha beta surface of integrins that bind ECM proteins. And then finally, people would say, well, these are not really 3D, they're 2D, they're not organoids, but we've coupled with click chemistry. I do not know how it works, other than that it's very fast. Invasin to a polyisocyanate gel created by our collaborator Paul Kauer. This now is a gel that's liquid at 0 degrees, a gel at 37 degrees. You can load invasin on it. It's reversible, so you can go back and forth between the different temperatures 10 times, and it'll still be a liquid at 0 degrees and transparent, and a gel at 37 degrees. And we can grow organoids in here. They really nicely polarize compared to BME, at least as good as Matrigel BME in 3D. And again, the same reporter organoid now in 3D. You see the hormone secreting cells, the goblet cells, the enterocytes, and the occasional Paneth cell. I think with that, I have used up my time. I thank you very much for your attention, and I hope I left some room for Q&A, and I'll stop share. Thank you.
There we go.
Okay. Can you hear me now?
I can hear you, yeah.
Good, thank you. Well, I was saying that was a fascinating presentation. Thank you so much.
I was briefly worried that you had heard nothing of my talk, but it came across.
No, it was I love the animations. They're very helpful. I had one question about adult stem cells, and to what extent not only, well, you've demonstrated how you can coax them into specifying various cell types that you expect in the organ from which you isolated the stem cells, but is it possible to take an adult stem cell from one organ and coax it into being a stem cell for a different organ?
No, so even in the gut, when we take sort of proximal duodenum, which is the start of the gut, or distal ileum, there are some subtle differences in transporters, for instance. Even after a year in culture, they still know exactly where they came from. So they're really dumb. They make exactly the tissue where they sat in. Also, therefore, very safe. The only way, so we've not been able to push them into any other fate. So within their lineage, they can make everything. But they can, so for instance, all the cell types that I showed you, but a proximal duodenal cell has a very hard time to make an L cell because they sit in the distal part and they make a lot of Greyland cells, the enteroendocrine cells. So, and the only way, for instance, to turn a gut cell into a stomach cell is by knocking in or knocking out transcription factors. But because then you get closer to iPS cells, I guess. So as long as we don't touch the transcription factors, they're really stable.
Question in the back?
Yeah, I have a quick question. So thank you for that great talk. Hopefully you can hear me over there.
Yeah, I can hear you.
So you showed us some great evidence that the LGR5 positive CDC cells are the intestinal stem cell. And yet there's some evidence in the literature that if you ablate these cells, that they're not completely necessary for the intestinal regeneration, let's say. And there's been a couple of papers recently in Cell that suggests maybe other cells are involved. In addition, I just wondered if you have any thoughts on this topic.
I have very strong thoughts on this. So we actually were amongst the first to show that when you kill these LGR5 cells, they get immediately replaced by cells that normally like fated enterocytes that are enterocyte precursors. If you left them alone, they would just become enterocytes. But if you mark them and you then kill the stem cells, they will turn around, go to the base of the crypt, and become multipotent. And we've shown this, several other labs have shown this, and this, so it's very different again from what the hematopoietic stem cell field has taught us. So we see the same thing in other tissues as well. There's very broad plasticity in these stem cell systems. The liver, I don't think that people believe in the evidence that there is any real liver stem cells, but they're the hepatocytes or the cholangiocytes, the only two real liver cells, endodermal liver cells. They will just simply revert to a stem cell state, make everything, and then go back to what they used to be. So there is a lot of plasticity, which explains many of the phenomena. Having said that, the two Cell papers, to be honest, don't provide any positive evidence for something that's higher up than the LGR5 cell, because that's what they state. And if there would be a cell that's higher up, so the quiescent stem cell that everybody's been looking for, you would see. So if we mark, and this is a bit of a complicated argument, but if we mark an LGR5 cell, the entire crypt eventually becomes blue. If there would be a cell higher up in the hierarchy, a year later, that blue should be gone, because then that LGR5 cell should have been replaced by something higher up. That never happens. So that really never happens. And so there isn't, as far as we can tell, we've looked extensively for 15 years. There's nothing higher up in the hierarchy than LGR5 cells. But LGR5 cells, we call it the everyday stem cell. But almost every other cell in the crypt can take that role if the stem cells are gone. They basically just go down, they end up in the crypt base, which is the niche, and then they become LGR5 positive, and they start working like the everyday stem cell. And I believe that many other solid tissues do the exact same thing.
That was a great talk. I just have a quick question, because my lab showed are responding from the stator neurons to regulate taste tissue stems homeostasis. I'm just wondering, the gut system, where's our responding from come from?
Yeah, so our spondin is not produced by the epithelium, that's clear. The Paneth cells make wind. But the arspondin comes from the stroma. There are several different labs. We haven't really worked much on these subepithelial layers. They're not endodermal. They are mesodermal. They're called telocytes sometimes, or myoepithelial cells. So they seem to be a source, but also the blood vessels and also the thin layer of muscle, the muscularis mucosae, seems to make arspondins. So there's actually circulating arspondin in your blood. So there's a lot of, and there's 4 arspondins.
All right, Dr. Cleavers, I know it's dinner time there, so I'm going to let you go, and thank you again for such a stimulating talk. Thank you.
Yeah, enjoy the remainder of the symposium. Thanks very much for having me.